the last part of the program. So we have Lloyd Johnson, who was mentioned a few times here. It's payback time, so you could hit him a few times now, Lloyd, it looks like. Okay, so we have Lloyd here. He is the Senior VP for Pharmaceutical Research, Development and Operation of Selecta Biosciences, one of the startups that has been mentioned before. Lloyd, like the others, also have been involved in many other startup companies and took things from the initial invention to the phase 1 through registration, so his experience is extremely wide. He was professor before that also at chemical engineering at the University of New South Wales in Sydney, Australia, and he received his BS in chemical engineering from Queen's University, Canada, and his MS and PhD in chemical engineering from MIT. So Lloyd, looking forward to your talk.
The right slide. Perfect. Thank you. When Omi was talking about conviction, a phrase I often use with my team, they come with a new idea and what they want to do, I said, will you bet your paycheck on it? If they say yes, we go. If they say, often they say, well, let me look at it a little bit more, but they usually get there, and that's where we move forward and we make a lot of good progress that way. So I'm going to talk today about Selecta, where we're actually developing nanoparticles for immunotherapeutics. So you've heard a lot about cancer applications, oncology applications, a little bit on the cardiovascular side. Here's actually where we're trying to interact with the immune system. We basically melded recent advances in immunology with advances in nanotechnology to do this. And over the last 15 to 20 years, there's been a lot of advances in immunology, understanding it both at the cellular and the molecular level. So vaccines overall, when we talk about vaccines, when I talk about vaccines, you all think of your flu shot typically, right? Vaccine, I'm going to make antibodies, prevent you from getting disease. And those are the classic sort of prophylactic vaccines that everyone's familiar with. They prevent disease. They're they avoid very significant healthcare costs, so it's actually very important in terms of managed care and other things to get people vaccinated to prevent future diseases. So we work on those. We also work on therapeutic vaccines. And again, I'm a chemical engineer. I'm not an immunologist, but if you go back to hundreds of years ago, your immune system is your body's pharmacy. There weren't drugs, right? So you got sick, your immune system fought it and you lived, or you died. And then herbs and things and early pharmaceuticals started helping either boost the immune system or actually more directly attack things. So here we're going back to try and harness the power of the immune system to treat disease. So we can place the need for cross-economic therapies. And so there is actually one therapeutic cancer vaccine, Provence from Dendron. It's a dendritic cell vaccine. It's personalized, but it still is immunotherapeutic. And then this last is a very new area that we're working in, which we call tolerogenic vaccines. So one example of that is if you get an autoimmune disease, for a reason nobody knows, your body starts attacking itself. The only therapy today, other than managing things like TNF-alpha levels for rheumatoid arthritis, really for many autoimmune diseases, the only thing you can do is do global suppression of the immune system, giving systemic steroids or something else. You do that, then you're susceptible to disease. What we call tolerogenic disease, or what's called antigen-specific tolerance. So what we do is we re-educate the immune system. So we use a nanoparticle essentially to send a message. Yes, I guess there's a drug inside it, but really what we're doing is sending a message or a blueprint to the immune system to reprogram it to do something. And in the case of tolerogenic vaccines, we're telling, for example, in type 1 diabetes, for reasons people don't know, your body starts attacking the beta cells in your pancreas. I don't have type 1 diabetes, my body ignores them. People who do. So what we do is we give a nanoparticle to the patient that then basically re-educates its immune system to say, you know what, ignore those are good guys. Ignore those, and then that helps cure the disease. But keep active on everything else. I won't talk about this much today, but it's a very interesting area that we're working. So overall, just on our general pipeline, one of our therapeutic vaccines it actually makes antibodies, though, is against nicotine. So if you smoke, nicotine goes in your lungs, the blood, goes your brain, hits acetylcholine receptor, you get dopamine, you feel really good, so you keep smoking. We do is with nicotine, we actually create nicotine-specific antibodies in your blood, so when you smoke, the nicotine now is grabbed by that antibody, and this antibody-nicotine conjugate, that's too big to cross the blood-brain barrier. So it keeps the nicotine from getting your brain, you don't hit the dopamine, you don't hit the acetylcholine receptors, you don't get that rush, and so now it's easy to stop smoking, but more importantly, most people quit for a day, a week, a month. What happens is, bad day at work or something, you smoke one cigarette, Acetylcholine receptor, dopamine, all right, and they keep smoking again. Now, if you have the vaccine on board, you don't have to do anything. You don't take pills every day. It's just on board, just like your flu shot. Now, if it binds it, you don't get that euphoria feeling, so therefore it's easy to stay quit. And that's currently finished a phase 1 trial, and we're moving forward to another trial, which actually being done in Russia. We also have a collaboration with NIH on malaria. Malaria has been a big problem. On cytolytic, we'll talk mainly today about our cancer work, our cytolytic T cells. We have a lot of work going on here in the U.S. We also have a grant with the Skokobo Foundation. I talked about our type 1 diabetes supported by JDRF. We have a large collaboration with Sanofi in the area of food allergies. Food allergies are just an unwanted immune response. An allergy is an unwanted immune response, just like an autoimmune disease. So we're basically trying to re-educate the body to avoid that. And then anti-drug antibodies are also a big issue. For example, you're getting protein replacement therapy, for example, hemophilia. If you don't have factor VIII, you bleed. You give factor VIII, but if I don't have it, my body sees it as a foreign protein. The main thing your immune system do is react to foreign proteins. So many people who have this disease, they react to factor VIII, they make antibodies to it, so next time they get an injection, it doesn't work. So we look to reprogram the body so that it ignores those. And that's true of many biological drugs. It's actually becoming a big issue in a number of areas, these anti-drug antibodies. Just when you give a biologic, as you know, there's many biotech drugs out there, your immune system actually often recognizes it, has a response to it, which then can affect it the next time it's given. So you've heard a lot about these oncology drugs, and they're basically fighting nature. Okay, we try and work with nature. So your body's immune system has evolved over millions of years to protect you against pathogens. Pathogens, by and large, are nanoparticles. So viruses are nanoparticles. Flu virus is about 80 nanometers in size. They go down to maybe 20, 30 nanometers. They go up to about 500 nanometers, depending on the virus. Bacteria go from a few hundred nanometers up to a micron or two. So if I want to give an oncology agent, so we haven't talked about long-circulating nanoparticles, what are they trying to evade? They're trying to evade the immune system. So I'm trying to do the opposite. I'm trying to actively engage the immune system. So I'm trying to work with the body, work with what your body wants to do to particles, and nanoparticles in particular. And we work in this typical range in here, around 100, 150 nanometers, which we find to be a very good range. It's very highly recognized by the immune system. So this is a stylized version of a natural pathogen, happens to be an influenza virus. And this is what we call our targeted synthetic vaccine particle. So a virus has a number of different characteristics. One of the main ones is what's called the B-cell antigen. So on the surface of a flu virus, you have hemagglutinin protein and neuraminidase protein. This is what hemagglutinin mainly is what you make when you get a flu shot. You make antibodies to the hemagglutinin. Similarly for the self-assembly technology that Omi talked a little bit about using those amphiphilic polymers. We can array on the surface of our nanoparticle, so we just don't make nanoparticles, we're making a nanostructured particle. We can array on the surface of the particle the B-cell antigen. Again, we can control the density to be the appropriate level to get the maximum immunogenicity. The main thing here, well, before I get to that, the T-cell antigen is that other proteins here engage something in your immune system called MHC class II. This gives something called T-cell help. If you don't get T-cell help, you don't get a strong immune response. Similarly, we can encapsulate, in fact, we've discovered a number of very short peptides that we can make synthetically that basically universally can engage T-cell help for people. The main thing your immune system does is ignore things, because if it didn't, we'd be a big mess. And basically, allergies, immune reactions, you don't want your immune system. You probably ignore over 99% of what you breathe, what you eat, everything else. But your body's actually there's single-stranded RNA inside of flu viruses. And this engages in something called TLR, toll-like receptors, in fact, toll-like receptors 7 or 8. These TLR agonists are basically molecular machinery in your body that looks for danger signals, or so-called adjuvants. So it tells if it sees single-strand RNA, it says, this is a problem, I need to fire up my immune system because there's a pathogen attacking me. Similarly, bacterial DNA is very rich in CG, and so-called CPGs, and because of that, if you see bacterial DNA, you also fire up TLR9. So that's basically why you react to pathogens and not to many other things. Similarly, inside the nanoparticle, we hide it away. We have an adjuvant that then gets released once the particles are taken up. And then this is a nanoparticle because it's a lipoprotein shell. And then as many others talk today, we use the polylactic co-glycolide, the PLGA, polymers, these break down in the presence of water to lactic acid and glycolic acid, which are then naturally cleared through your body. We also use PEG in the formulation. So how does this work on the immune system? So we give our particles sub-Q. Our target, which when I first started with Selecta, I didn't understand this is what happens, is that basically your immune system happens in your lymph nodes. It's the main place where your immune activation actually occurs. So we're actually trying to get it to the lymph node. So we give a sub-Q injection, it flows to the lymph node, and that's what this circle here is. And once it's inside, the particles do a number of different things. They can get picked up by subcapsular sinus macrophages, which hold them for B cells. So now a B cell, so what I have on the surface of the particle is something, for example, I want to make antibodies to. And you have millions of B cells in your body that all have sort of different shapes. And so something, if I'm trying to make antibody to this water bottle, something that's got a shape that's not exactly it, but close to it, that'll bind loosely. So now that it's bound, it internalizes it, it releases the adjuvant, it activates the B cell, and it also takes that T cell help antigen and presents it on the surface. At the same time, the particles are being picked up by dendritic cells, which are, also present this, this peptide to engage what are called CD4 help cells, and this T cell now helps the B cell into a germinal center. And what happens here is that the particles are held there by the follicular dendritic cell, and this sort of loose binding, it's a competitive binding event. So if you bind, you get to reproduce. When you reproduce, you mutate. And over time, you get a very, very exact tight fit. As everyone knows, antibodies are extremely specific. So that's why they're used a number of different things, very specific to other, to what their your flu antibody attacks the flu, doesn't attack other things. There's another arm of the immune system, so that gives us these strong humoral responses, the antibodies. The other arm of the immune system is the cellular immune response. Again, in layman's terms, since I'm from Boston, basically what you do is you're arming cells and you tell it, go kill all the cells in your bodies that are wearing a red coat. You like the British, right? So we killed them. So here it's a protein marker. There's some protein marker on cells in your body that these CD8 T cells, which are educated by the dendritic cells, go out, these T effector cells go out and kill various cells. So we can teach this to go kill a cancer cell, for example, looking for a specific antigen on a cancer cell. Manufacturing, very important. I actually was, before joining Selecta, and I was the first employee of Selecta, I was head of manufacturing operations for Alkermes, which you heard a little bit about. We made thousands of kilos of polymeric nanoparticles. We had large plants. I built large plants, ran large plants. In vaccines, you don't give very much. You don't give it very often. So this is our phase 1 vessel, which is small. My favorite Diet Pepsi can, which I drink too much of. But this small little vessel here can do up to about 10,000 doses of a vaccine. It's about a 10-gram scale. So this is the emulsion vessel, and we go through a very simple process of doing an oil-water emulsion. That amphiphilic polymer is moving around inside that oil droplet. Just like, for those who are not familiar with the emulsion process, it's just like your oil and vinegar salad dressing. You get the oil droplets inside the vinegar. That oil droplet is where all my polymers are. This amphiphilic polymer is moving around in it. The hydrophilic end gets near that oil-water interface. It'll stick in the water phase. Now it has inertia, it doesn't move anymore. So we enrich the surface of the particle with whatever on that hydrophilic end, which then lets us make that nanostructured particle. And then basically we dilute it in further amounts of water. The solvent runs out of the particle and hardens the nanoparticle in that shape. And then we use very standard standard equipment like tangential flow filtration and other things from the protein industry to clean up the particles. We make them, just as Jeff mentioned, we sterile filter them so we don't have to do an aseptic process up front. I've done aseptic particle processing because the Alkermes products are about 100, 150 microns in size, and it's a real pain. Actually, the biggest advantage of nanoparticles over microparticles, and that has a huge advantage, is that nanoparticles don't settle in liquid over any time of interest. So I can sterile filter them and do a liquid fill. If you're doing larger particles, like microparticles, they settle. So you can never get an isotropic sample using a liquid filling device. So you never get content uniformity. So what you actually have to do is do a sterile dry powder fill, which is extremely hard to do, and there's very few people in the world other than Alkermes that can do that. So it's a very flexible, modular application. We have B-cell antigens, T-cell antigens, immunomodulators, and nanoparticle polymers. We'll go through some of these with some examples. So in the B-cell antigen, we've used proteins, peptides, sugars, small molecules, put all of these on the surface through various versions. This is a small molecule small molecule antigen. So if you give a small molecule, your body actually doesn't make any immune responses to small molecules. because it can't do what's something called crossing the B cell receptors. So we give that even with an adjuvant, which fires up the immune system, nothing happens. Now if we take that same small molecule antigen, put it on the surface of a particle, and give it some T cell help, okay, it gets some sort of reaction, but without any adjuvant, it doesn't really work very well. Now if we put the adjuvant inside, we get now, this is a much, 2 orders of magnitude increase. So basically what happens is a log scale of antibody response, antibody type. This is the level of the antibodies. Over here is a viral peptide antigen, so instead of a whole protein, we just take a short peptide out of it. If you give it, again, it's too small, even with a conventional adjuvant like alum, it's too small to actually make a strong immune response. If we put it on the surface and we give it a good adjuvant on the inside, what happens here is you get some response, but because this is a short peptide, there's no T cell help. So without the T cell help, you don't get a strong immune response. Now we put the whole particle together and we get, again, 2 orders of magnitude increase. So the whole TSVP can give very significant increases in the antibody levels. Immunomodulators, we use TLR agonists on the stimulatory side, and different tolerogenic agents on the tolerant side. So this is a protein, it's a full protein now, long protein, and it's not a very immunogenic protein. So if you did this with flu protein, this would be a really high bar, because flu protein is very immunogenic. This protein is not flu. It's not very immunogenic, though it's one of interest. And you can give 100 micrograms of protein plus adjuvant, and you get a titer of only around 10,000. If I give only 5 micrograms of the protein inside a particle, I'm going up 1 to 2.5 to 3 orders of magnitude. And this is a different formulation, which has 2 micrograms. So 50-fold, 20 to 50-fold lower amount of the protein, what we're trying to get antibodies to, when getting 100 to 500 times higher antibody levels. And one of the key issues here is, so let's talk about it. What do you often, when you get vaccines, you get flu-like symptoms. These are actually often caused by what are called systemic cytokines. So on the left here, here's our particle with the antigen on the surface and T cell help inside, but no adjuvant. And it works a little bit okay. It looks a bit like a virus to your body, so you get a low immune response. If we take this particle and we mix it with an adjuvant, a TLR agonist, we get it works. Adjuvants systemically mix, and all the current vaccines that are approved used adjuvants that are systemically, just admixed with them. We get an order of magnitude increase, so it does work. But here now what we do is, it's the same adjuvant, we take 1/10 the amount of adjuvant that we gave free, and we put it inside the particle, now we get an order of magnitude increase. And then over here, these are systemic cytokine production. So TNF-alpha, this can cause aches and pains, IL-6 can be responsible for fever, and the colors match. So if you don't give any adjuvant, you get no systemic cytokines. But if you do admixed adjuvant, you get a huge spike in these things that cause flu-like aches and pains and fever. Here, despite the fact we got a 10-fold increase in the antibody level, so it's 10 times more effective, we actually basically have almost no levels of systemic cytokines. So we keep all of that in the local lymph node. Now in the local lymph node, it's reversed, because cytokines are needed, because that's how your body's immune system, it's the language. Cytokines is what is how it communicates. So in the local lymph node, we get very high levels of interferon gamma and IL-12, which then promote this strong immune response. And this is really because we're targeting the particles in 3 ways. They have the right size, so upon injection they flow to the lymph node. Once they're there, they have the right characteristics that they're picked up and look like a virus to your body, so it's picked up and acted on the immune system. And thirdly, the particles, the adjuvant inside the particle is actually triggered to release once it's inside of an antigen-presenting cell, and therefore we don't get this systemic exposure. So a nanoparticle polymer, and I think Omid alluded to this, the PLGA nanoparticle system is very flexible. PLGA, for those who don't know, it's the same material that's in resorbable sutures. It's been in clinical use for over 30, 35 years in improved products, and many improved products. But it's extremely flexible. You have the lactic to glycolide ratio, which changes hydrophobicity. You have the molecular weight. You have a number of different things you can play with, and it's a very, very flexible system to do a number of different things. And we use this in optimizing nanoparticles. So we design a formulation, the polymer composition, the antigen, what load, what immunomodulator, and how much of that. Once we make a nanoparticle, we do analytical testing. We look at its size, its physical chemical properties, and also what's important is that release rate, so how fast or slow you're releasing the adjuvant. And then we do some in vivo screening of select formulations, because we make a lot more of these than we can test here. And we look at either antibody levels, CTL, or inhibition. This is in the tolerance field, depending on what you want to do. And actually, we've come to, although we're not in the small molecule realm where you have structure-activity relationships, when I see this testing, I can tell you a lot about how it's going to perform here. So we know sort of what zones we need to be in. It's not just a blind gamble as we go forward, as we do new projects. So this is one where we're looking at in vitro adjuvant release. So we can make particles that release, this is the percent of the adjuvant that's released almost immediately, or over several days, almost a week out here. So we can make anything in between these. And here, it does have a biological effect. This is an increasing adjuvant release. So this is a very slow release on this end. And you go through to get to a peak. And then as you release very quickly, it doesn't work so well. So we actually try and work in here. So it's important to be able to tune that release. So this is now looking at cancer vaccines induce antigen-specific CD8 T cells to actually attack tumor cells. And we did a number of formulation screenings here. So here, this is an interesting experiment where what we did was we created some cells, some that were pulsed with an ovapeptide, which is what we're vaccinating the animals against, and some that were with just a control peptide. This is done in a, this is done ex vivo, and you've got, so you've got these cells. Then what you do is you inject those cells into a mouse that's either immunized or a control mouse. In the control mouse, basically nothing happens to the cells. You basically get the cells the same. They're diluted a little bit, but you get the same proportion of control and the target cells. But in this mouse, all the target cells are completely eradicated. So basically this mouse has CD8 T cells against this ovapeptide, and when you put them in the mouse, those CD8 T cells go in and kill all those cells. So we completely eradicate them, but we don't touch any of the non-target cells. So this synfecal is the actual peptide sequence, but here, when we make a particle that has ova and adjuvant inside it, we get this big upregulation of the CD8 T cells. If you put the adjuvant in a particle but not the protein, you don't get anything. And if you put the protein in a particle and give free adjuvant, you get very little, maybe doubling, but very little of this. So you really, the key of the nanotechnology here is how it actually is inducing these very strong CD8 T cell responses that you don't get otherwise. So this is a typical T cell response that you'll see in the literature. And in fact, when it's a typical nanoparticle, or well, in this presentation I'm calling formulation 1. This is formulation 1, is what it gives. It rapidly expands over about 7 days, then it rapidly contracts down to about day 17. What we want to do is have sustained CTL activity. Because basically after 17 days you don't have many of those killer soldier cells going out trying to kill things. Right? So we're trying to sustain that. So we made a bunch of different formulations, and I've shown whatever that is, 8 here. We probably made 50 or more. And we tested them, and you can see some actually on day 17, which is out here, where it's low in the standard sense, are actually pretty high, sustained. And so then we selected one of these, and this is sort of the, this is actually the formulation we optimized for antibodies. And when we used it in the CTL, we see what pretty much everyone sees in the literature, this rapid contraction. When we took the optimization of the polymer and everything else to do it for CTLs, we actually can get sustained very high levels of CTLs out for 21 days, which is actually pretty important in terms of trying to attack cancer. And some of that has to do, we believe, with how it's doing the antigen processing. So this is an interesting model molecule called DQ-OVA. It's when the protein is folded, it's quenched. But once the protein gets clipped, it gets processed, and that's what happens inside a cell, an antigen-presenting cell. It chops it up into small peptides, which then get presented at MHC class 1. It now fluoresces. And so we put some of that part in a particle and give that to the animals. And the formulation 1, you get about some processing. On MHC class 1, about 13%, but we get much stronger processing in formulation 2, which is the one that has that sustained CTL activity. So we believe that this has something to do with inducing that very strong response. So now if you look at this, and this is, I'll go into some more disease-relevant models. This is just simple ET7 tumors here. So here we give the TSVP treatment, and this is the sham treatment, or the empty nanoparticle, is what was given here. And you can see that we get pretty good tumor suppression, and we get very strong CTL activity over the entire, up through day 25. So this is another survival curve, similar type of TSVP, not so much, but if we take these survivors, okay, so these survivors, they were given tumors, and they were given, then a few of them died, a number survived, And we take these survivors, we don't do anything to them. We just let them live, we don't give them any other medication, anything else for 3 months. And then we re-give them the tumor. And none of them generate, the tumor doesn't take. And basically you've got the sustained T-cell memory, so anytime the tumor cell comes out, or there's a tumor cell that circulates around, then these CD8, they're like sentinels going around, they kill it. So you cannot give these mice, even though we gave a pretty strong dose, you cannot give these mice the tumor again. And someone else mentioned about combination therapies, and I think from a cancer perspective, immunotherapy is probably best in the combination, because if you've got a large tumor, the immune system takes a while to get going. And sometimes you don't have a while, particularly if you're in the end stage of cancer. But if you do something like either surgery or radiation or some chemotherapy, or what Oliver or Bind and Jeff, give some sort of treatment to help knock down the tumor, but in combination give the immunotherapy, and again, chemical engineer, not the biologist, but in the simplified term, no matter what you give, surgery, radiation, chemotherapy, nanoparticles, if there's one cancer cell that survives, it'll become 2 cancer cells, then it'll become 4, and 8, and 16, and so on. Eventually it'll come back. So you want to have this sentinel that's going around, basically trying to keep your body free of that cancer. And so I think the immunotherapy can offer that in combination with some other cancer therapies. So this is then, we can also put in disease-specific epitopes or whole protein antigens, and this is a HPV model. So this is the E7 protein, E7 expressing HPV, human papillomavirus. So this particle that we're using here contains just a single peptide. So instead of the entire protein inside the particle, there's a peptide that's about 10 amino acids long. That's all that's in there with an adjuvant. And what you can see here is this is the empty nanoparticle, and then this is the HPV, the TSPV treated, and all the mice basically don't grow any tumors whatsoever. We also gave low doses and high doses, and what you do see when you look at the real baselines here, tumors do start to grow in a number of these animals, but then they get shut down. So even though they do grow, the immune system's able to overcome them and take them even at the high dose. You need to give both the peptide and the TLR agonist, so this is just the empty nanoparticles. If you just give a nanoparticle with a peptide in it, really no difference. If you just give an adjuvant, not much difference, but when you give them together, you can see this great reduction in the tumor volumes of all the mice. So this is compared to a DNA therapy. This is actually from a publication in 2009. This actually BMS through a company called Inovio licensed where this work came from. But basically it's the same study. It's 5 times 10 to the 4 cells. We had 5 out of 5 tumor-free mice on day 38. So they had 7 out of 10 on day 49, and 8 out of 10 on day 35 in terms of tumor-free mice. The surviving controls, none died on our controls. We had a stronger tumor line than they did, even though it's the same tumor. As you know, there was a variable. So on day 30, all the controls were dead. And didn't get out until day 49. They still had 1 out of 10 still surviving. We gave our treatment on 3, 7, 14, and 21. Theirs was 3, 10, and 17. We used peptide, their DNA, the same one. We had a adjuvant, and they did not. But we can do pretty well on those sort of things. This is also seen here in the sort of the overall survival, but also in the mouse weights. This particular tumor causes wasting, and you can see these very significant difference in the weight of the mice over time. And why is this? It's a specific cytotoxicity again that I showed before. So we get this long, sustained cytotoxicity compared to the others, as well as confirmed by ELISpot. And this is the same tumor line, so it's still the TC1 tumor line. It's a different, those were given, I believe, sub-Q or IP, but this is given IV for looking at a model of metastasis. And what you can see here is that the empty nanoparticle control group has significant, these are the lungs, and looking here, significant metastasis, lung metastasis of tumors on the lungs. If you look at, it's the same treatment where the treatment's given 3 days after, it's a therapeutic model after the the tumors are inoculated, you can see that the TSVP did complete protection over that same group. So we're a clinical stage company. We have looking at therapeutic nanoparticles for immunological applications. We use self-assembling, biocompatible ones. As I mentioned before, what we really do is provide information or a blueprint to the immune system to generate a desired response, whether trying to raise antibodies or CD8 T cells or trying to tolerize the system in a certain way. We can encapsulate the adjuvants so they target the draining lymph node and we don't get the systemic cytokine production. We get robust cross-presentation, which then gives rise to very long and sustained CD8 T cells. We've demonstrated protection, both EG7 and, more importantly, the HPV model. And we have successful GMP. We've made a number of GMP lots as well. I think that's it.
Very, very interesting. I think I don't know too much about this field, but I know actually in this building, there's a lot of people in immuno-cancer therapy. They set up a GMP also facility here. So maybe you'll end up collaborating with some of them. This is a very hot topic in the cancer field, and we appreciate sharing your experience with us. I saw June, right? Okay, yeah. June, maybe you can use a microphone. I don't know what happened to. I gave specific instruction. Nobody listens to me anymore.
I think I finished early.
I got a shout.
Okay.
So it's really nice. I was really impressed by the immunotherapy for cancer. So my question is, have you ever thought about combining personal medicine to this immunotherapy for cancer? For example, you get a patient-specific peptide based on their particular personalized cancer, and let's say you get a patient sample, do a quick analysis, you track the peptide and load it with your whatever particles and inject back. Would you think that would be the future?
So personalized medicine is I think there's some ways that it can be done. That's going to be a little bit more difficult, mainly because of regulatory and simple GMP reasons. So that if depending on how much time you have, but if you need to make a specific GMP batch of nanoparticles for each individual patient, that's going to be difficult. Now I know there's a group from the Weiss Institute at Harvard that they're doing something like that, but what they basically make from a GMP perspective, they make I don't know the details, I just sort of read, but what they make, it's Dave Mooney and Glenn Dranoff, and they have, I think they make a wafer that's basically a porous polymer, and so they make that GMP, and then what I think they do is they get the patient-specific tumor cells, and they actually just soak it at bedside, and it sort of absorbs, and then they implant that, and that does some promotion. So the nanoparticle, although very simple and straightforward, probably not at this point anyways to make an individual lot for an individual patient. And my personal view of a personalized medicine, although cancer may be a little bit different, is instead of being personalized, you're going to get kind of into binned medicine, right? So that, I have, there's 7 billion people in the world. I have trouble believing there's 7 billion types of breast cancer, right? There may be a lot of different types, but the thing would be to, bin you into one of 20 groups or something like that, or 10 groups, or whatever it might be, and a lot in the diagnostic field trying to figure out, which bin you work in the best. And I think many people in the cancer field have had very good results, and particularly the cancer vaccine field. And what happens, it works in some subset of patients, and go to a large phase 3 trial, although it still works in the subset of patients, they miss primary endpoints because that's diluted out by this larger population that doesn't respond.
Great point. Yeah. Oliver?
No, I first of all to agree with you that immunotherapy in cancer, I think, is the end game. So keep my fingers crossed for you because you'll be successful there. Then a quick question on, you mentioned on the manufacturing side that you can do aseptic filtration. If I understood that correctly, given your particle size, why does that work? How do you exclude viruses and bacteria, given that you seem to be right in that range where you would expect them to also be?
So sterile filtration doesn't take out viruses. You've got to keep those out otherwise. So sterile filtration is defined as 0.2 micron or 220 nanometer filtration. So our particles are small enough to go through a 0.2 micron, and that's acceptable from a sterility perspective. But of course, we take, the front end. We're not making it in somebody's garage. It's still, pretty nice, clean, but it's not aseptic. In fact, we use some. I've been a manufacturing guy for a long time, there's a real difference between 0.2 micron filtration and sterile filtration. So we use a lot of 0.2 micron filtration in the early parts of our process. We call it that because it's not sterile. To be sterile, it's got to be, you have to have a sterile vessel in the back end, which is where it gets more expensive than the aseptic side. But we basically try and keep everything out at the front end, use low end of tax and water, all that kind of stuff.
Yes, yeah.
From Ansani. I was wondering, you create this therapy, you educate the immune system to attack cancer cells or maybe other targets. But how about memory of the immune system? I didn't hear you talk about that. Can you more elaborate?
Yeah. So the memory, yeah. No, actually, what we're doing is creating central memory T effector cells. So I showed you those mice that survived, and then we didn't do anything to them for 3 or 4 months, and then we re-injected. So that's evidence, and we've also done some actual profiling, immunoprofiling and FACS analysis of the cells. We actually generate long-lived memory here. With particular formulations, other formulations don't do that.
Okay. I missed that.
Okay, great. So we thank you very much. It gives me great pleasure to introduce our last speaker, Greg Lensa. Greg Lensa and I met actually a long time ago. I have been very much, as well as a lot of us in the community of molecular imaging, and inspired a lot by the early work with Sam Wickline. Greg and Sam has really pushed the aspect of MRI molecular imaging early on with these delivery systems that increase the payload. So a lot of us have benefited from and learned a lot from this work. Greg has a background of a pharmaceutical company. He worked in industry, a manager of Monsanto, initially in St. Louis, where he developed a lot of work related to their preclinical development of Posilac. And then after he trained as a cardiologist, he serves in biomedical engineering at Washington University, as well as the Barnes-Jewish Hospital, where he practices medicine and cardiology. He is also working in the field of both cancer research and cancer nanomedicine, as well as cardiovascular disease. His MD is from Northwestern and PhD and MS in genetics from the University of Georgia, and BA biology from Colby College. So it gave me always great pleasure to listen to Greg, and thank you very much for accepting to speak here. Thank you.
Thank you. Thank you very much.
Started, and this is, we can go from yours or from here?
I can go.
Oh, yeah, I transferred it
Okay,
earlier,
no
right?
problem.
Okay, and you can move it from here.
Okay.
Thank you very, very much. It's my pleasure to come and chat with you a little bit about what we're doing. We work across a lot in cancer, both in solid and liquid tumors as well as arthritis. Today I'm going to mostly restrict my comments, since it's a pen, to cardiovascular disease and pulmonary. As you said, I'm a cardiologist, and one of the things I have to do is see patients in the emergency department. And the reality is that some patients come in just like this, and it doesn't take an Einstein to figure out what's going on. Their EKG, everything tells you, go to the cath lab, and we send them. But the reality is that a large majority of patients come in with chest pain, and we don't know what it is. We can't determine it, and the risk and liability of sending them home is onerous. So we admit them. And we take serial telemetry and serial enzymes, and then if we rule them out for a heart attack, we go and do another stress test. Families are ajar for a couple days trying to figure out dad or mom having a heart attack, or they don't know. People fly in from a long distance in case this is the big one. Very much a problem, especially when 85–90% of them have nothing to do with their heart. So one of the things that we're using in medicine a lot is cardiovascular CTA. And it's quite good for ruling out disease, but it's really not that well reimbursed. And the problem is what the FDA wants is for you to determine that a person has disease. They want you to target it. So some of the agents that we currently use, our iodine blood pool agents, won't help us do that. And there's a variety of agents I might mention. This was a particle that was developed by the Harvard group, Weisslater Lab. It's really something you dig out of the ground, and it's bismuth sulfide, it's an ore. But you can see that it's very dense, and you can see it here in the case of this lymph node, but it's big. In fact, if you give this to somebody, it will never leave their body. It's for life. So that's really probably not a starter for heavy metals. Because in the end, you can't really prove that it's going to be safe 20, 30 years from now if they have something else go on. It's how do you do it? So you're in a tough spot. We work, one of the areas I wanted to chat with you was with spectral CT. This is an emerging modality. It's a development that's gone on in the last decade, and it's a technology that really springs from CT being attenuating imaging, like X-rays are, white and black and all the grays in between, to more of energy resolution. And we'll talk more about it. But the bottom line with spectral CT, and this is our instrument here from Philips, is that it allows us to go ahead and possibly take a CT angio and then put on a hot spot where our particles, for instance, targeted fibrin, would recognize a thrombin. And this could be done in the emergency department very quickly. You get the diagnosis, you got fibrin in your coronaries, you probably need to go to the cath lab. You don't, you probably can go home and see your doctor, and maybe, you know, we might give you something for spasm. So the long and short of it is, this could be paradigm changing when we're able to do this. Essentially, the holy grail, whether it's spectral CT or however it's done. Now, why can't you use regular CT? And the reason is because CT sees attenuation. The detectors just basically see the integration of all the energies of the photons. And because of that, because our coronaries and our carotids are so full of calcium, it's very difficult to target a CT contrast agent to any of those tissues and recognize that it's not calcium. And in that situation, especially in the coronaries, you're stuck in a situation like this, where you get a CT picture where you see all this bright stuff. You get a K-edge picture, which we're going to talk about, that's really to the targeted fibrin, and you can separate calcium from fibrin. That's the goal of spectral CT in the context of coronary. So what is K-edge? So some of you probably aren't familiar, but you may remember in chemistry we had these various shells, and the K-edge is really related to this K electron in the first shell, and every element has one. And each one at every element has unique energy. So what happens is that if you had a photon come in from an X-ray beam, and it's at the same energy as that electron, the two will fuse. And when they fuse, you get this. It's a discontinuity. You get an increase in absorbance. And so these energy resolving detectors, which are different than the detectors today, they're able to count photons, and I'll talk to you a little bit more, but basically they can count photons at this side and determine that they're low energy, and count photons on this side that they're high energy. So the K-edge detection or K-edge imaging is really a function of this discontinuity. Now, every voxel in a CT picture is really a summation of three things: a photoelectric effect, a Compton effect, and the K-edge effect. All those things can affect attenuation. In a normal CT scanner or X-ray pixel, we just pile them all together and you see the net effect. But in these high-resolution energy resolving detectors, they're trying to bin out the energies. And they get, through mathematical processing, a photoelectric effect, Compton effect, and K-edge effect. Essentially what this allows them to do is have the CT picture, the normal one you see, and a K-edge image. And because they're all done at the same time, they're superimposable. So it allows you to see that fibrin on the calcium. The calcium has a very low K-edge, around 4. It's below the bandwidth of an X-ray beam, it absorbs nothing, and it doesn't show up in the K-edge image. So through this differentiation, you can separate things like calcium and a contrast agent. Now this work was done right here at Mount Sinai, of David Commode, Zahi, and others here, Liam, and you can see that they used their HDL particle and they entrapped gold. Gold particles, particle in a particle. And what they did, and I just wanted to point out, was show using essentially the same machine as ours, but done in Hamburg, that you could take iodine, which is here in the blood pool, and separate it from the macrophage imaging they had done with their gold particle on the uptake with CT when it's done with spectral CT. So they have the regular CT, they have the blood pool image with iodine, and they have spectral CT imaging with the gold. They see all of it instead of it just being black and white. We also made one, and it had been used originally for photoacoustic tomography for sentinel lymph node imaging, and I just wanted to point out that it also brings in a strong signal. But the other main point I wanted to bring here is, as we develop these kind of agents, you can only go so far with the chemistry. Because the other half of the equation is really on the instrumentation, signal to noise, and the post-processing. And I think it's quite clear, you can see that the signal to noise, or the contrast to noise, here in an iterative reconstruction, where this is back projection, is much greater. The noise is lower. So as we go through, and especially on things like CT imaging, there's a need for both chemistry and imaging to work hand in hand. And that's the relationship we had with Philips, and still do. Now our approach to this problem, the broader approach, is actually to use soluble metal, organocomplexes. Why? Because they can leave the body. We need to have metal in, metal out. That's my opinion. I think it's a lot easier at the FDA level. Now, it puts high densities of these metals in these particles. We're talking about 40, 50 weight percent, okay? But they're all small molecules. And this one happens to be incorporated in a phospholipid-coated particle and targetable. And we did the first question we had, because we went through 4 generations of these particles, and we published this in New Chem, was can we even see this thing? Because when you're targeting a surface, as opposed to that sentinel lymph node, or even the macrophages where things are accumulating and concentrating, it's very, very tricky. You have very little thin layer having to create a big signal. And so we did this study in rabbits using an antibody, and we demonstrated that indeed these high-density bismuth particles, that we could see them. Now that vessel right here, that's a 1.4 millimeter vessel. That's actually a small coronary size. Done in a rabbit, and you can see it's well differentiated from the bones. Now, one of the things I wanted to point out in the case of this, which is fibrin imaging, is that this fibrin all through the plaque, you can see it here in this carotid endarterectomy specimen, is purple. So it will not behoove us to detect fibrin in a plaque here that's unrelated to a potential rupture or clot formation in the lumen here, because these plaques are going to rupture and heal and rupture and heal. So one of the advantages that particles can help you in this regard is that they can target the fibrin in the surface and avoid the particle accumulation in the plaque wall. And that's important. If you're going to make a decision to send someone home, you don't want it because they had fibrin in their clot. Fibrin clots in their plaques. Now we've gone on, and our scanner is very slow. And we've done these systemically using a phage display peptide to fibrin. And this is an example where we give it systemically, put them in our little scanner, But unfortunately, our scanner, it has a 10-minute to 15-minute slice time. So it's impossible to keep a rabbit alive, let alone even keep a dead rabbit, you know, from decomposing by the time you get this. So what we do is we take the vessels out. So this is a polycarbonate phantom. This is 1 centimeter. And these are clots that were created by Rose Bengal in the lumen, targeted with fibrin systemically. And I think it's quite clear. You can see very high resolution imaging of these very tiny clots. And they were targeted systemically, so this is our goal. Now, for those who are interested more about the topic of spectral CT, I point you to this special edition coming out in January, I think, Contrast Medium Medical Imaging. It has a paper from this group here, David Camone, Zahi, and others, and also ourselves and other top groups. And it'll tell you about the background behind spectral CT, the applications behind it, and also about a variety of different contrast methods that are being used to explore it. And I suggest that if you're interested, this will get you 90% to knowing all there is. Now, we do a lot of work in angiogenesis, and we don't use peptides generally. We use this quinolone that was developed by DuPont Medical Imaging. DuPont Nuclear Imaging, and then became part of Bristol-Myers Squibb Medical Imaging. And we couple that to a linker which we put into the surface of almost all the particles we make. And when they're about 300 to 500, in this case on a perfluorocarbon particle, you're talking about binding of 50 picomolar. That's essentially stick and stay. Now, the important thing that's important to realize here, and this is not true of all the ligands to ABB3, is what does it bind? And what you can see here is that these rhodamine particles are in this Matrigel mouse that has transgenic LAC-Z-TIE2. You can see that the PCAM, which is a marker for endothelium, and the particles line up precisely. But when you look at the TIE2 image, you can see that this is not mature. TIE2 is a marker of a vessel where the endothelium has been polarized. And you're starting to form pericyte smooth muscle cells. And I'll show you what those look like. If you look here in this part of the vessel where there's no targeted binding, you can see that there's endothelium and TIE2, no particles. So you have to understand what it is you're targeting. And this is what that looks like in Matrigel. So this is a technique called photoacoustic tomography, where you put light in and you have sound out. And that's all I'll say about it. But essentially it allows you to see red blood cells and contrast agents that are designed for photoacoustic tomography. The long and short of it is, in this picture at baseline, you see forming vessels. And you see some red blood cells coming through. But if you look down here, several hours later, you can see there are bridges and sprouts and all kinds of things coming on here, like here, where the arrows are, that are not seen here. So what happens is ABB3 is actually in vessels that do not even have full connection or any blood flow yet, as well as vessels that are just formed where the endothelium hasn't polarized to have an apical and a basal part. And that's why ABB3 can be targeted there. And as I showed you, it does not target to microvessels. So this allows us to characterize what we see. So in the context of atherosclerosis, angiogenesis is a major component of the arterial wall. It's feeding the plaque. It's feeding the macrophages, T cells, and other cells that are making up the stroma of the plaque. And it's growing literally from the outside of the vessel in. And I'll talk a little more about that. Now, Renu Vermani, in a paper in New England Journal 7 years ago, showed that one of the sources of intraplaque hemorrhage, which I'm going to talk about in a moment, is angiogenesis, and maybe it's the source of intraplaque hemorrhage. So, how do you determine what's a vulnerable plaque, and how do you intervene on plaques that might actually rupture in the near term to prevent strokes or heart attacks? Well, today, the best marker, in my opinion, is intraplaque hemorrhage. And in these 3 studies, you can see the intraplaque hemorrhage patients, and these are patients who had mild, moderate disease, carotid disease. These patients had poor event-free survival over a period of up to a couple years. Okay, so in other words, if you have intraplaque hemorrhage and moderate carotid disease, you had, in this case, 50% chance of having a stroke. This case, 65. This case, 80-ish. I mean, well, actually, it's this difference. 80% of the people didn't have a stroke, and 20%, let's say, did. And here it's 50-50, and here it's, say, 30% had a stroke, 70% didn't. So it's a high marker for a stroke, and if we could image that on every person who comes in the emergency department who had some symptom that sounds like a stroke, but they don't have overt symptoms of neurologic dysfunction, might be good, except for the fact that what you really want to do is take them for carotid endarterectomy or a stent if they're in jeopardy of having a stroke. And you can't take patients on a 50-50 basis. Gee, you had some symptoms, I see some iron in your plaque, I'll flip a coin, because you have a 50-50 chance at best of having a stroke, and accept the risk. That just doesn't fly. Because the problem's so severe, we've gone to taking patients who are asymptomatic now with 70% disease. And I don't know how long that's going to last. So intraplaque hemorrhage is a start. It's driven by angiogenesis. It's one of the precursors to it. The question is, what more can you add to that to make it so you can detect whether someone coming in with symptoms is at high risk for stroke, and is it going to come back 2 to 6 months later or a year later with a stroke? Well, one of the areas that we've been interested in for a long time, of course, is angiogenesis in plaques. And we showed that we could image angiogenesis in plaques, which is challenging in and of itself, using MRI at the clinical field strength. And this was one example of a study that we did where we not only imaged it, but we put drug. In this case, fumagillin was an anti-angiogenic drug that was really from the Folkman lab, originally, Don Ingber. And you can see that the amount of angiogenesis that we see in MAP in these fat-fed rabbits, and these were about 100 days on lipid diets, targeted with drug really knocked down the angiogenesis. And let me just point out that here's a plaque, and here's the adventitia, and here's the expansion of the neovasculature in that adventitia, where the plaque is, and not over here, where there's no plaque. So there's a good correlation between this. This is very early. The thing is, 30 micrograms of fumagillin is a trace dose, homeopathic dose. It's 10,000 times less than the dose that was used for cancer. The doses that were used for cancer were actually led to neurocognitive dysfunction. So one of the things nanoparticles do in general is help us to get more of the drug where it needs to be and lower the dose, and make something that was essentially toxic relatively safe. And in fact, in the studies we've done with this, the drug effect is negligible. But you can see that it has a significant effect on angiogenesis, and that's corroborated through histology. So that particle, actually a slightly different version with a different chelate on the side, which was gadolinium DOTA here. That particle had this one, which is DPPA, went to the clinic. And in the first couple doses, patients started to have some problems. Their problems were transient back pain, very transient, but unexpected at the dose we had. So we put a pause on that study. We went back and we analyzed their serum, we analyzed everything, and basically found out that when we made the transition from gadolinium DPPA BOA to gadolinium DOTA, which was designed to hold the gadolinium better to prevent what was then nephrogenic systemic fibrosis, not that we would have had it, but it was a fear. We gave a particle that now greatly induced complement activation. And you can go to this to get more details, but you can see the activation here with DOTA. And let me just point out, that doesn't mean that GAD-DTPA is safe on particles. And the reason is that the GAD-DOTA was on a PE backbone, just so you know, and that puts it out in the water, which gave us much more relaxivity. But the GAD-DOTA, the GAD-DTPA BOA is really nestled at the phosphate head. So it may not be available in the same way as the GAD, but if we had made GAD-DTPA PE and done this study, might have had the same problem. So one of the things we did is we had a problem. We went back to the drawing board, and we approached it through two ways. One was make new particles that don't have that problem. The other is, what can we do about the GAD particles we're already making? Is there a way to go about it? Because these are perfluorocarbon particles, and I'll just bring that up in a moment. So we started looking back at our friend manganese. For those who aren't familiar with MRI, basically we're looking for our unpaired electrons. And so in gadolinium, you've got 7. And in the case of manganese, you've got 5. So it's the next best. And we used manganese oxide, which are particles, and did the particle-in-particle thing, which was kind of like what they did with HCl, although it was at a different time. And the result was that we had pretty good relaxivity. But in the synthesis of manganese oxide, you form manganese oleate, which is an organometallic complex. We used the manganese oleate and used that in a particle, and we had much better relaxivity. So the first question you might ask is, how is it possible you're getting all this relaxivity, that is signal, from a particle where the metal is on the inside, and it's not iron? And the answer is that the manganese oleate, and the reason why on an equimolar basis it works this way, goes all the way to the water level. It leaks all the way up to the phosphate head. So inside the outer layer, there's a heavy density of manganese that's having a paramagnetic effect on the circulating protons near the particle. And in the case of the manganese oxide, they can't penetrate as close, because the acyl groups of the lipid prevent it. It's as simple as that. The signal was very good, but we were interested in angiogenesis and have, in the more recent years, looked at adding gadolinium, because when we took the manganese oleate, as good as it was, great for fiber, not enough for angiogenesis. Too low. You're in the no nanomolar level. So we made gadolinium manganese oleate, essentially putting gadolinium on the outside. We still had to consider the NSF part, so we stuck with the DOTA. And what we did was we dropped the level. As we dropped the level of GAD-DOTA, we found that actually, as you'll see, even at 0.6 mole %, we had very high relaxivity and synergistic. And I'm not going to go through all the MR for you, but essentially, the manganese substituted, if you will, for the fluorine, plus the gadolinium had a synergistic effect on the relaxivity, greater than the manganese plus the gadolinium by themselves. And this is a CH50 study done in vitro with sheep red blood cells. You can see that if you look at normal saline, there was no increase in complement activation compared to, say, the DOTA. And here, if you do it in mice, looking at C3A like that Western I just showed you, you can see that there's much more, and almost negligible here. Essentially, it's negligible. So what happened to the signal? Well, the manganese oleate particles by themselves have a signal, and they tend to maximize itself at around 10 nanomolar. And the manganese oleate with the GAD supplementation at 1% shifts it way down here into the 5 nanomolar range. And in that range, this little difference makes all the difference in seeing angiogenesis. And once again, you can see the time-dependent increase in neovasculature and mapping to angiogenesis in these atherosclerotic aortas. Here's the signal change. So the point I wanted to make is we now have a way of going back using a paramagnetic agent, T1 agent, and mapping angiogenesis. So what do you want to know about angiogenesis that would be additive? And one of the things I think that's important, and I bring it up here, is that it's these red spots, which are actually over 4 standard deviations from the background, versus these blue, which are 2, that we're interested in, and where they occur. In other words, where are you having this hot angiogenesis? And where is it occurring relative to the plaque lumen, the endothelium covering it, the intima? My point is that if it's occurring very close to the intima, like in this case, where the angiogenesis and the macrophages are right here, for instance, in the plaque where this is the lumen, that's probably very vulnerable, and especially if it's associated with intraplaque hemorrhage also. So that kind of information is totally different than if all the angiogenesis is back here. That's a progressive disease, no doubt, but it's not nearly as, let's say, vulnerable. So I think it's this kind of information that will help us to make judgments. Now, originally, I thought that the answer here would be to go to carotid endarterectomy or do something to stabilize this. But one of the things that has happened is that I'm working in other areas, and I've learned something about fumagillin that I didn't suspect. I knew that it had an effect, and I knew that the effect in this particular study, where we did it in those same kind of animals, lasted about 3 weeks and then went away. And if I had that in conjunction with statins, which are a normal therapy for patients with atherosclerotic disease, that we could extend that effect. So in other words, we had acute treatment that we could keep going with a chronic therapy we're already using. That's a great clinical translatable thing. At first I thought this was due to the hydrophobic nature of the drug. And it was just lasting 3 or 4 weeks and then it just all run out. But now I know, and our data will, we've worked this all out, the whole pathway, is that our anti-angiogenesis is directly to the endothelium cells. It's not anti-VEGF or some indirect measure. And now we know that when we give fumagillin, for instance, that it actually changes the inflammation in, for instance, the synovium of arthritis. And it changes the phenotypes of the macrophages and it changes the production of cytokines. And it's actually anti-inflammatory as well as anti-angiogenic. So you don't only have the disruption of the matrix, the gruel that angiogenesis will do, occurring right under the cap, which itself is probably a weakening thing, but it actually may medically be stabilizing the plaque by stabilizing the macrophages. And we worked out the entire pathways for this, and you'll see it. So the other thing I wanted to point out to you, and this is one of the few cancer studies I'm going to show you, is that it's important in some diseases to know about where things are, like in atherosclerosis. I'm really not that concerned from a ruptured plaque standpoint if all the angiogenesis is in the far wall of the coronary or aorta or carotid. I'm very concerned if it's right under the intimal plaque, right on the low shear side. In cancer, it's not such an issue. It's almost really a matter of the percentage of neovascular expansion you have associated with the tumor that's related to the growth and effect of the tumor. At least that's what we've found. And to make a long story short, what's important about all these type of things is that they have to be robust. So I'm a clinician, right? And so I fundamentally need a number, not a picture. I need a number that says this is a 2, this is an 8, this is a 15, okay? And then a set of guidelines to tell me, okay, the 2 we wait, the 15 on the border, maybe you do something, the 8 you do something or not, the 15, treat. We can't get along without some quantification. So quantitative imaging is critically important. Also, it needs to be robust and repeatable. So you see in this particular rabbit, this area here is this area here, and this area here is that area there. Okay? Now back to the fluorine particles, you really don't need the gadolinium, because advancements working with Philips led to the ability to do dual imaging of fluorine and protons simultaneously. And now that's potentially available on a clinical machine, if you're nice to them. It's certainly on our 3 Tesla. And you can see that the images we were getting with the T1 agent are the same as we get with the fluorine. The difference is fluorine's more quantitative. Yeah, this didn't pack. So essentially one of the issues is motion. You have a fluorine image and you have a proton image. But what Philips has been able to do, working with us, primarily by themselves, but working with us, is to take the proton image to register the fluorine image. Because I said it had to be quantitative. And you see here, where there's no motion, we have these phantoms. You can see down to 4 nanomoles of particles. But with motion, which isn't showing up, you get this blurring. But you can use the proton image, since they're exactly at the same time. It's broadcasting 2 radio stations at once and hearing 2 radio stations at once and deconvoluting it. You can get right down here where you compensate mathematically by gating. And so now all of a sudden the proton image is locked in, the fluorine image is locked in. Now, one of the things that we wanted to do was look at asthma. And the reason is we were learning about, over the last couple of years, about the anti-inflammatory effects of fumagillin and anti-angiogenesis, as well as by the fact that it's a serious problem in the patient populations we see. I just pointed out here, 56 billion in 2009, and it affects everybody. And in a way, this air bronchus reminds me quite a bit of what's going on in atherosclerosis, except they don't have as much of the remodeling that we do because of the architecture of the cartilaginous architecture. So we did our particles in asthma. In this case, though, it was in a pulmonary model of inflammation. It's called pulmonary arterial ligation. And we wanted to see, could we even see these things? Because at the point is, you know, MR and lungs, they just don't go together in general. Why? Because, you know, there's motion, there's not many tissues, so there's not much tissue, so there's not many protons, and there are chemical shift artifacts. Add them up together, it's just not pretty. But fluorine is not dependent on proton, right? It's independent. So we said, let's see what we can do, and maybe our anti-inflammatory effects will have an effect. So you can see that where we did this, which was very near where we ligate the pulmonary artery surgically, that we got a lot of inflammation right around the pulmonary veins here, and you can see it all co-marking between the red particles and the green lectin. And here in the lungs, we also found it was occurring in the outer distal parts of the lung. Now, I'm not 100% what this is, but what I think is you have to realize that's going on here physiologically is we've stopped blood flow from the right heart, okay? And as a result, there's a shunt from the bronchial arteries, which is where the angiogenesis occur, to the venous system. So it has to shunt back. You need blood to get around. So this pulmonary artery ligation is going to create a certain amount of early angiogenesis as well as vessel dilation first and then angiogenesis to increase the blood flow through the bronchial artery back down through into the left atrium. And that's what I think this is all is happening here. So here we have red for the particles, green for the lectin, and it's kind of yellowish if you see where they're all superimposed. So this is the image. And this is the whole image. You can see that near the hilum, where we had the most angiogenesis, it's pretty dense. And then here in the more apex, you can see more of the angiogenesis occurring in this part of the left lung. And you can see that a lot of these particles just accumulate in the spleen and liver. Yes, the left lung was more. Certainly, we've targeted it to the The ABB3 targeted is much higher fluorine signal than the non-targeted. In the competition, the non-targeted sham. So I wanted to talk a little bit about drug delivery. Now, all the people have talked about is putting drugs in the middle, and those familiar with us should know that we don't do that. We put our drugs in the outer membrane. And when you put drugs in the middle of a particle, you can leak them out in the blood, but that's not targeted delivery, that's delayed, that's just controlled release. You can get them into a cell, but then they have to get the drug out, and that was pointed out earlier too. So our thought, right from the beginning, was the idea of get the particle to touch the cell and deliver the drug, and not have to have all the losses associated with getting in and then getting out of some vesicle. So we call that contact-facilitated drug delivery. You can see it here with the particle binding this cancer cell, and you can see the rhodamine lipid, which is in the outer part of the particle, streaming all over and through. And this is the picture that was taken by one of our colleagues at WashU, showing the particle fused to the outer membrane of a C32 cell. And some work that's described in these papers shows that the drug actually goes passively from the outer membrane of the particle to the outer membrane of the cell. It's actually, the energy comes in the curvature of the particle. And then it goes with ATP from the outer membrane of the cell to the inner membrane of the cell. And as I'll point out, the inner membrane is contiguous everything in the cell, but the mitochondria. So we thought we had actually a good system, very holding on the drug in dissolution. The fumagillin was hardly released, maybe a few percent locked in plasma. But in vivo, when we did mass spec, tracking the homing ligands, the fluorocarbon with GC, the drug, and gadolinium, everything was sticking together except the drug. The drug was leaking out fast. This led us to have to find a solution. So one of the things that you do through the process is you think you've got everything solved, well you don't, because some problem's always around the corner. We tried many, many different ways of putting drug on there and having, enzyme linkers like esterase and you name it on the outside. Those didn't work. Why? Because oftentimes you can bring the drug to the target, but the drug isn't bioavailable to the target. So just because you can stain a drug or do GC analysis or mass spec on something in a tumor or in a plaque doesn't mean it's actually available. And that's one of the things we found out about this. So we put the drug down here and created this whole family of prodrugs, in this case with fumagillin. And in the case of fumagillin, which was photo unstable, we eliminated that too. The drugs are delivered when the cell membrane binds again with the lipid membrane. You have delivery, and then here cytosolic lipases release it. Now I pointed out that if you give free doxorubicin to cells, and these are endothelial cells, the drug will go right into the nucleus. But when you give the prodrugs, they're going to be all through the cell membranes and then released. And I didn't have time to add it in, but you can do fluorescence lifetime imaging and literally track it from the particle to the membranes, to the cytosol, to the nucleus. And I just want to point out that when these drugs are in that membrane in this way, even though there are lipases in blood, they're stable. So in this case, here's the drug. This case is docetaxel. And here's docetaxel, this whole prodrug by HPLC. Here, we're in plasma, we're spiking with extra lipase. Nothing. If you go ahead and you add the alcohol to crack the emulsion, then it exposes the membrane back into the plasma and the lipase, it starts getting broken down. And if you actually track what's being broken down, you can find actually that it's being broken down just like docetaxel would. So just point out that we made a lot of prodrugs, the fumagillin. We made the prodrug, the camptothecin, which also helps stabilize that. We've made a variety of them, even methylprednisolone. We're working a lot in the area of transcription factors. And docetaxel also. So basically, I just try to give you an overview. I try to keep it into cardiopulmonary. And tell you that there are a lot of different modalities for molecular imaging. There's spectral CT, there's photoacoustic, there's MRI. There are new contrast agents being developed for all of them. And they all have been considered in terms of their safety. So there are ways to get around it. You can get metallized. You can get high reactivity without gadolinium. And there are different ways to put drugs. In our case, they all have lipid particles, although we do make polymer ones. They all have phospholipid membranes. And so all of them, the ones I showed you and some of the ones that we made that are only 16 nanometer, are all biocompatible with these drugs, which lock the drug in, take it to the target, and then release it. And that lowers the dose. So in the case of fumagillin, the savings, the efficacy we could replicate with only 20% of the drug that we used, which was already 10,000 times less than the drug that's used systemically. So there's great opportunity to make safe, effective drugs. And even though we all thought, in this room I'm sure, that the only thing that anti-angiogenesis would do was to change the blood flow characteristics, further work in all these different areas reveals that we don't know that much. And now we understand that it's highly anti-inflammatory. And that was what was giving us the response times that we see. So you really need to stick to something and keep digging on it too, because you'll be amazed at what you learn when you dig down. Just want to recognize the whole group here, Sam, of course, and all my collaborators, and Ocean Nano and Phillips. Everything I've shown you is licensed. Some of it's still in the clinic, some of it's just starting. Few of the drugs aren't licensed, but basically everything is translatable. So if you want to talk about the issues that were associated, I'm available.
Thank you very much, and this was, as always, fascinating. A lot of interesting ideas and concepts that you presented to us. Maybe if people have questions in the audience. So maybe, what if?
Are you guys still pursuing these molecular imaging agents based on gadolinium in terms of trying to get that clinically, or?
The answer is yes and no. So with the high gadolinium, no. But with the manganese oleate GAD, that's licensed out for that purpose. Because the complement activation. And you can't go back to DTPA. It just won't work, especially that kind of DTPA, because it doesn't bind the GAD that well.
All right, Kevin, and then Walter.
Could you explain the synergistic increase in relaxivity that you talked about?
Yeah. So I didn't want to go through it too much, but essentially what we did was we took manganese oleate particles with the same amount. We took GAD manganese oleate particles. We took GAD-only particles and compared all of these and found that the relaxivity is more than additive, considerably more than additive. And we used the gadolinium at 1.25, but found that actually at 0.6 even, which was our range that we tested, we had essentially the same relaxivity. So the manganese oleate provides a lot. The low level of gadolinium by itself provides practically nothing. And the GAD manganese oleate is much more. If you take manganese oleate particles with the GAD oleate particles, same concentration per voxel but separate particles, there's no difference between that and manganese oleate on T1 relaxivity. Yeah, so there's a lot of stuff that I didn't go through, but that's how we came up with the synergism. And to be honest with you, I was a little unexpected.
So what's happening? What's causing that?
I think what it is the manganese oleate creates a dipole. And then when we put the gadolinium DOTA on there, we get a marked increase enhancement of that dipole effect. When we use the same amount of gadolinium with gadolinium DTPA-BOA, where the gadolinium is nearer the manganese, which would have an enhancement if it were fluorine nuclei, it has no benefit. It increases it, but instead of being 1%, you have to use 10%. So I think you have a dipole. This is my theory, not necessarily fact. You have a dipole effect from the manganese olates you don't get with the fluorine. We know that the relaxivity improves when you take the gadolinium off the surface. We published all that stuff. And I think what's happening is we're having a synergistic effect by being just out there in the water, still within the dipole. But I leave it up to the physicists to someday actually work, if that's actually the cause of it.
Kevin, we're waiting for you on that.
Zahi can figure it out.
Yes.
So you talked about the complement activation, and you said that with the manganese oxide, you had plenty of access to water, but it was tucked down closer to the particle surface. I was wondering, is there a way to do that with the gadolinium as well? Can't you engineer the surface?
Well, there's two things. So if you make gadolinium oleates, it's a wax. So we did try that. It's just not really formulatable. But also the situation is gadolinium oleate isn't that stable. And the third thing is that at the time we were doing that work, the whole idea of gadolinium itself, especially in Europe, was an anathema. So we tried to stay away from it. The complement activation is a function of what's on the surface of the particles. So that's how we get it. I didn't say one thing. Everybody keeps talking about complement activation as though it's alternative pathway. It's primary classic pathway that's amplified by the alternative pathway, and that's all published in that JBC and in another paper that's coming out. So we actually have immune antibodies that are recognizing these particles. They recognize PEG, they recognize a lot of things, and they're activating the complement pathway.
So, Greg, maybe I'll ask you a question. You mentioned some of the drugs that you're trying to encapsulate or to add transcription type of drugs.
Yeah.
You mentioned. Can you say a little bit more about
sure.
that?
I didn't include it because we're working on transcription inhibitors, and primarily a couple of them now. The one I showed you there was MIC-MAC, and we're using it in myeloma. Very effective, more effective than the drugs. In general, transcription factors are relatively poor solubility, very bad kinetics, and in some cases can go to, if you give them systemically, they'll affect every cell, right? Okay, to make a long story short, by making the prodrug, we markedly prevent it from going to any other cells, but also we activate it more, and we bring it right into the cell. So it's not only more potent than the parent drug, so like the doxorubicin was somewhat similar. The difference in potency of the prodrug for MIC is maybe 4 or 5 orders of magnitude than the MIC drug itself. And by the way, these aren't real drugs. These are, some of the things we work with are drugs that are approved. Some of the things we work with are new drug entities. So this is a new drug entity where we synthesized it. But it was discovered by someone else, but we synthesized it and make these things. I think it's actually better than trying to stop a pathway. That's why I went after it, because in a pathway, there's always a way around it. It's like you can't, it's like whack-a-mole, but they all have some common places. So we're working on different transcription factors to block the whole damn chain going down and then affect all the genes. So in the case of MIC, to give you some idea, it affects 25,000 genes. All right. So when you're having a MIC inhibitor onto a cell, it has pleiotropic effects, so long as MIC is activated.
Okay, Greg, thank you so much. So we're coming actually to the end of this, and maybe to make it simple for you on a Friday afternoon, we're going to eliminate the panel discussion unless there is really objection coming in from here. So one of the things that's always good is to start a meeting on time, but to finish early. It makes it actually a great meeting. So before I finish, I want to again echo the thank you. We had all the support from people who helped us put together the program, Bob Langer, Omid Farshad, William Mulder, but also the administrative support and help and energy we got from Ina and Ayana. So I want to take some time to applaud, to thank them for all the support here. Also want to thank you for all the speakers who came and stayed with us, like Greg, till the end, and your participation. So we'll try to repeat maybe this event sometime in the near future. I would love to hear any feedback you have, any ideas you have in terms of pushing this, things we did not address. For the trainees here on campus, if you get stimulated by starting up a company, go talk to Willem. We'll try to talk to Omid. We'll push you in the right, push you in the right direction. So thank you very much.